मेरी आवाज आ रही है ना? सेकंड
Yes, ma'am.
আগে পাঠিয়ে দিয়েছি লিপিকা ম্যামেরটা ওখানে নেই এক্সেলে মেলে এমনি পাঠিয়ে দিয়েছি
meeting is now streaming live on YouTube.
Hello.
We have 4 more minutes to go, so
Okay.
4 minutes we can start.
Okay.
Good morning and welcome everyone to our webinar today. And we are going to give participants 3 more minutes to join the webinar from the start. So I would request everyone to mute your audio during the presentation. So we can start the webinar now.
Yes.
So welcome everyone to our webinar today. We are so glad to have you join on 14th September for the second webinar of the Drug Discovery webinar series with Professor S.G. Prakash Vincent presenting Transgenic Physics in Drug Discovery and Opportunities for Entrepreneurship. Thank you for joining us for today's KTBI Interactive Live Webinar. Introducing myself, I am Elena Das, working as Lead Analytical Facility at KIT Technology Business Incubator. Along with me, we have Mr. Saikat Gupta, Associate Design and Campaign, TBI. So, as all of us are aware, zebrafish are cost-effective alternative to some mammalian models and fastens the drug discovery processes, and therefore has immense opportunities for startups and pharmaceutical firms targeting the transgenic or non-transgenic fish-based drug discovery. In the drug discovery webinar series, in today's webinar, we'll get a deeper understanding of how zebrafish serves as a tool for drug discovery and is a prominent vertebrate model for disease and discuss about entrepreneurial prospects. And in order to make the sessions interactive, today's 1 hour 30-minute session will be divided into two parts with a 1 hour of theory or presentation session by Professor Prakash and followed by 30 minutes of question and answer session following it, for which I would request all participants to type your questions in the Zoom chat window or unmute yourselves during the question and answer session and directly interact and ask your questions. For which, however, to avoid any overlap while questions are asked, I would request participants to type your names in the chat window so that the questions can be taken serially. And the webinar presentation will be shared after the webinar, and the video shall be shared within 1 week. For availing the e-certificates, we will email the feedback link to participants who have opted for it while registration, and after filling the feedback, you will receive the e-certificates within 1 week. Now I would like to welcome our esteemed keynote speaker, Professor S.G. Prakash Vincent, sir. Introducing sir. Sir holds the PhD degree from three institutes, University of Madras, University of Zoophysiology and University of Bonn, Germany, and worked as postdoctoral fellow at EMBL, Germany. And Professor Vincent is Chairman Associate of BRICS Biomedical Consortium since 2014. He is also focal point for European Commission Horizon 2020. He is a DAD Fellow Germany and EMBO Fellow, Marie Curie Fellow, and UNESCO Biotechnology Fellowship awardee. Currently, he is professor and HOD of Center for Marine Science and Technology of Sunderlal University. Recently, his group has discovered a novel antibiotic which is active against human superbugs. Now, I would request Professor S.G. Prakash Vincent, sir, to kindly enlighten us on today's topic.
Good morning to all of you. It's a great pleasure to stand before you to give a talk on transgenic fish in drug discovery and opportunities for entrepreneurship. Thanks to KT TVA team and Alina and all the CEO and all the members for inviting me for this talk. Good morning, participants. Just to see the content of my talk, it will be like a drug discovery in overview, zebrafish medaca, why it's a good model, advantages. Then coming to techniques, I'll just cover few techniques first. Gene knock-in, how I could develop a gene knock-in model of medaca at Heidelberg. Then Insitu hybridization, Antisense RNA-based knockdown, then stable gene knockouts. That's a technical. And I'll just move on to transgenic fish application in drug discovery. How human cancer mimicking zebrafish are made, organ toxicity, angiogenesis, finally into high throughput screening. Final section will be entrepreneurial aspect. I'll be telling you what is heat and how valuable it can be, and like a novel, this the therapeutic tracks, and mainly targets, novel targets, and finally into what we are planning to do with the technology we have at Transgenic fishes. Now, giving an overview, you see this slide. This is a total overview of drug discovery. From target discovery, you can see assay development, screening of hits to leads, lead optimization, development, then clinical phase. You will be aware, many of COVID vaccines are in the clinical stage study. So one drug to come to market, it takes almost 10 to 15 years, and about $2.8 billion. In Indian money, it's about 80,000 crores. So lot many funding and lot many investment is there for a drug to come to market. Here you have to have two things in mind. We all know small molecules hits, but one of the main thing is targets. Okay, the important protein molecules in a human body where a drug can go and bind and bring down the effect. Now, zebrafish Daniero and Medica, another Oriceus latipus, is used as a model organism, and every month or every 6 months, lot of literature is coming on using zebrafish, the transgenic zebrafish in drug discovery. And this zebrafish can is used mainly from target discovery to assay development, then all this screening of hits to leads, then a potency, dose response, counter screens, selectivity, mechanism of action, lead optimization, then finally toxicity testing, etc. So in the drug discovery process, the importance of zebrafish is getting day by day. This is the content of my talk. Okay. Now, why zebrafish? If you see this picture, zebrafish has 72% of orthologous genes as of humans. So if we see that this is the comparable figure, which can tell human brain to a zebrafish brain, cardiovascular system here, okay, liver here, okay, pancreas, okay, here, gallbladder, intestine, and the nervous system over here. If you see the zebrafish, you should think the size of the embryo is a pinhead size. The size of the pinhead. Okay. Within this thing, in 3 to 4 days, total organs, all these representative organs develop. So you know that it can be directly placed under an objective, objective of the microscope. And once you label it, then you can see the organogenesis in the zebrafish. There you can bring a lot of information for screening. So this is the advantage of zebrafish as a vertebrate. You have 72% orthologous genes. Orthologous genes mean almost only few amino acids change will be there, but the gene will be there. If you take a transcription factor or a neuropeptide, in human you can find the same gene in zebrafish expressed. So that is the advantage. Okay. Then this slide gives a lot of, we have flyers models, zebrafish, mouse, rat, etc. This is a little bit older side, but now if you see that this 4D imaging, zebrafish is one of the top. Okay, that I'll come in a few slides. What is this 4D imaging? And transgenesis is also zebrafish. If you now if you put this slide, it should be 4+. Okay, because this is done few years back by this. It's also published in Nature Medicine by Rishke and Curie. And that I took this slide. So lot of advantage zebrafish has as a model. And also it's a human anatomical system. It has a representative organ which we can play with the different tools of transgenic tools. Now this is a work like a gene knocking, which I try to develop. Or not try to develop a transgenic Medica. This is a counterpart of zebrafish. Only thing, the good thing is that it doesn't have pigments. It's very clear. You can visualize the transgenic organs in this fish, and in Whitworth laboratory in Ember, when I was there a decade ago, so many, maybe 2,000 tanks are there with the Medica. Now, what I did to develop a transgenic, one was a gene knock-in procedure I applied to develop a transgenic fish. So, gene knock-in refers to a genetic engineering method that involves the insertion of a DNA sequence information in a genetic locus not found within the locus. So if you see this protein, okay, this has a gene, okay, normal. Now you see this has the same protein. This protein has a gene here. Now we have put a small another gene. This is the gene for GFP, okay, or green fluorescent protein. Okay, so you can knock in this gene along with a known gene. So along with this protein product, this gene also will be expressed. So what I did was I developed a transgenic fish which can express GFP and RFP in the cell membrane and also RFP in the nucleus. So I'll just go through the technical details of this methodology. So transgenic fishes like construction of vectors, microinjection, production of transgenic fish, fold imaging, confocal imaging. Okay. So now, as I told you, two proteins, genes for protein, GFP and RFP were taken. The advantage of GFP is it can absorb this 488 nanometer wavelength light and re-emit as 440 in the green fluorescence. So it will absorb the light and then re-emit as 440 nanometer, give a green color. Same way, a little modified one thing is RFP, which can absorb as 540 and re-emit a red color. So that is the characteristic features of this fluorescent protein. Originally, it is from jellyfish. It's not a fish protein or human protein, originally jellyfish, but used in thousands of laboratories for transgenic works. Okay. So what we did was we developed two vectors. Okay. So like Isaac-1 vector, we took the RFP gene, okay, and fused with the alpha actin. Actin, you know, actin and myosin are this membrane, cell muscle proteins, okay, which is expressed in all the muscles. Alpha actin is another form of actin. So normally a gene will have an upstream elements called enhancers and promoters. So that is taken and fused to the RFP, okay, and also a nuclear localization signal was put here. Then another vector, alpha actin enhancer and promoter of alpha actin with a GFP, a plasma membrane localization signal is put. Okay, so two vectors were constructed. If you want to go details of this thing, you can go to Grabber and Whitboard. Highly efficient zebrafish transgenesis mediated by meganucleus. This is almost like 15 years old technology. Now latest technology come, but because I did it very much practically, I'm telling it, and for knock-in, this is one of the best for like integrating the gene into the genome. So now, like the muscle cell, what happens is once if the vector, the DNA is integrated, it will produce the RFP, okay, and then anchor in the nucleus. Okay, same way, this gene, which is into the genome of this medaca, will produce the this thing GFP and anchor in the plasma membrane only in the myocytes, okay, or muscle cells, not everywhere, because this enhancer and promoter is active only in the muscle cells and myocytes, not in other cells, not in nerve cells. Though it is there, gene will be there, it won't be active. Okay, so this is the principle. So now, like once you have the vectors already, it's a molecular biology work getting the right vector. Then you collect the eggs of the medaca, and that's in the water here. Okay, you keep it nice so that the fastly the cell will start dividing. Because the day before, if you put the female and male and start mating, and the next day morning by 7 o'clock you'll see clutches of eggs. So you can collect the eggs and bring it to the laboratory. Okay, and put dice. And this tube has this vector, and plus meganucleus and enzyme. Okay, so you have to mix here and the micro inject. You can think that it's like less than the size of a pinhead, custard seed, like, very small and transparent, where you can see the blasto disc, okay, under the microscope, and then inject a small, like a smaller liquid here. Okay, so this is the process of micro injection, and you can arrange the embryos in an agar strip, because it's in water. It will move. So for micro injection, it will be like hold it tightly. So this is arranging going on. Okay. Then you have to prepare the micro tip needle. Okay, needle. then allocate a small vector, and then fill the needle here. Okay, nail the needle which is ready for microinjection. And now the needle is mounted to a small robotic arm. Okay, then through here you can see the tip of the needle and also the embryo, and through a pressure here, you can just dispense a small, very small aliquot, like less than 1 nano, like a microliter or less than that. Okay, into the embryo. Then you have to allow the embryo to develop. Okay, then you will see that after few days. Okay, few days, the embryos develop, and under UV, if you get a green light coming from the embryo, then you know that the GFP gene has been expressed, your vector has integrated into the genome. This is an F2 generation where we get a stable transgenic. Normally, you get a mosaic in the F0, F1, F2 you have to take, then a few embryos will be transgenic. Then you never need an injection. You just breed these fishes, you'll get a lot many transgenic embryos. Like if you have 10,000 embryos, just go on, keep on breeding this fish. Okay, then it can use for further studies. So this is the work which I did in developing a transgenic fish, Medeca, or the methodology, the basic methodology. Okay, so now this is one myocyte. If you see that muscle cell, you see the GFP, green fluorescent protein, being expressed, and RFP only in the nucleus. Okay, this is one of the pictures I took in this confocal. So you can zoom under the thing. You can have the zebrafish in a small petri dish, like in a water media, so that it will be developing there. Okay, so it's a live fish for even 2 days or 3 days. You can have it by using trichane. You can see that it doesn't twitch a little bit even, so that the plane of imaging will be there. So in the confocal has a good advantage that you can get a 3D imaging. Okay, normally you get a 2D imaging. If you go depth by depth, you can get a 3D imaging. Over a time, like every 10 minutes you take an image. Okay, then you get a 4D image. Like a movie, organogenesis, like a movie you can make. Okay, yeah, under the microscope. So this is one advantage of zebrafish vertebrate model. Never you can see an organ development. Normally in a mouse and all, you cannot do that. Okay, so this is the advantage that zebrafish has. Organogenesis, you can make a filament. Then you can intervene with any chemicals or anything for finding out which is the best hit molecule, or do some gene manipulation for finding the target. So this is next is a technique called Insitu hybridization, where like if you want to find out like where is the particular mRNA expressed, okay, which tissue is getting, because we have like 20,000 genes or 25,000 genes, and even like in humans, 3,000 proteins function is not known. 3,000, and zebrafish gives a good model. Take the homolog gene and then do an in situ hybridization. You can find out whether this gene is expressed in the eye or the muscle or the brain. So here, this is the technique. You take like any GATA, it's a stem cell marker. This is expressed in the stem cells. Take this GATA gene and then do an by clone the vector, which produces an antisense RNA. Okay, produce an antisense RNA. Label with a marker, with a deoxyribonucleic label. Then once you this is a whole mode. Then once you treat this antisense RNA, labeled antisense RNA, it will penetrate. These are dead animals. This penetrate and go and bind to where there's the original RNA. The GATA will have original RNA. It will go and bind to this RNA and make form a hybrid. So exactly a cell only producing GATA or expressing this GATA1, this antisense will go and bind. Then using an enzyme staining technique, you can visualize where is this decoration labeled probe, so that you can visualize. So like this, in situ hybridization gives the first hand idea which tissue express which type of mRNA. Okay, so that next is one technique called antisense based gene knockdown. So here they use a synthetic RNA molecule called morpholinose. Okay, gene knockdown by morpholinose, antisense. Chemical structure part of morpholinose is given here. So if you see this picture, morpholinose, so short segments which can go and bind to ATG of a start codon. of a particular mRNA, or in a splice site. You can chemically synthesis and then inject. So if you want to see one expression of whether a calcium ion channel is important for heart development, if you want to see, then you make an antisense for this protein, or the exactly which can bind only to this expressed protein molecule. Then inject into this embryo with a dye, just to see that how much penetration is there. After what happens is this will block the expression of this calcium ion channels. Then, if there is a phenotype difference, okay, so there is the heart doesn't beat normally, or 20% beating is there. Once you see these embryos under the microscope, you know that this gene has impaired the heart beating. Okay, so this is a technique also to find out gene function. Okay, so you can imagine how many probes can be developed, what are genes and how it functions in a phenotype, can be done by antisense gene knockdown approach. This is a transient, only for short time, maybe a few days that will be there. Then automatically the inherent system will take care, and then you'll have a normal heart function. Okay. Another technique is called gene knockout. Okay. Here we remove the gene from the genome itself. Okay, genome itself, so that a stable knockout can be produced. So, tremendous application in biomedical field. Like we can find out which are the proteins very much important for organogenesis, heart development, muscle development, for brain development. Okay, because they have the similar analogs. To human gene. Okay, now here this is a new technique 2018 you et al. In development cell, where single guide RNA, they took a gene, okay, had 4 RNAs developed, RNA developed from this gene, and then pooled and then injected into the embryo along with the Cas ribonuclear protein. You could find out that the total knockout of this gene could be obtained by using this technique, and there are different versions of CRISPR-Cas knockout. This is the standard tool for knockout in any model organism, and very easy to follow and use also. And these tools, as I told, gene knock-in, knockdown, knockout, okay, Insitu. These tools are used for doing lot of findings using zebrafish in drug discovery, which we'll be doing. Okay. So now, coming to the next aspect, what are the different areas where scientists have experimented and found out novel small molecules and the novel targets will be my talk. Cancer, angiogenesis screening, T-acute lymphoblastic leukemia, finding out a novel drug. Then zebrafish with the human cancer. This excited me actually. The zebrafish with the human cancer genes and screening. Okay. And personalized cancer treatment. Okay. These 4 I'll be covering on zebrafish as a model in oncology or cancer drug discovery. Then zebrafish used in epilepsy, kidney regeneration, heart repair, cardioprotectant, even diabetic drug screening. Okay. And finally, this is also a recent study, which is also very important because zebrafish used as to find a novel target for one of the sarcoma. Okay, because as I told you in the first slide, target discovery and hit discovery are the major things in a drug discovery. And a target is the most important thing. Now, if you have a very good target, then a lot of hits can be experimented with. So a disease-specific target. So if you can make a molecule bind out this target, then the disease should ameliorate. So that is the principle. Okay. So this is one of the best, the initial paper which has come on using target discovery using transgenic zebrafish. Now angiogenesis. Okay. Now you see that this slide, a tumor cell creating its own blood supply. Okay. So this is what does in cancer. No? When the malignancy goes on, this to the next nearby vessel, lot of blood vessels can be connected and make the suck the all nutrients and make the tumor grow at a very malignant way. Now, if you can inhibit this blood vessels forming, in a way you can kill this tumor. So this is what being done in one of the model of zebrafish. If you see this is comparable ISV, intersegmental vessels. This is comparable to this blood vessel. So any molecule which can inhibit this vessel in zebrafish also can work in a mammalian model, also finally maybe as the drug can work in the humans. So this is one area where zebrafish is being used. And next slide will give you like this is a robotic screening they have done. They made a transgenic zebrafish which has a FLI like promoter fused to a EGFP, enhanced GFP, which will be expressing in this promoter driven area, and this using like lot of scannings and high throughput assay. Finally, the knowledge generated during each prior classification step enabled rule to set a specifically quantify IFP. So the computer system is developed, softwares are developed. It can specifically quantify IFP data, which comes from the zebrafish. So hundreds of thousands of zebrafish can be embedded in the 96 well plate, and then drugs can be tested, like 1,000, 2,000 drugs can be tested, where inhibition of this ISV is taking place. So then they can take some lead molecules. So this is one phenotypic based screening done in zebrafish. Now another one is exactly how they came out with the drug, which can which is used screen for it's for acute lymphoblastic leukemia. It's a leukemia. So here, this leukemia allows the T cells to develop very fast. Okay, or it's a T cell specific leukemia. So what they did was they took a zebrafish and then made it transgenic with a LCK promoter and EGFP. So now, if you see that the T cell area only gets the green color, because this is very specific to only to T cells. So now this is a model. This is a model they took for screening. So all zebrafish will in this well will have a small area which is green in color. Okay, so you know that this is developing the developing T cell. Now they screen different drugs. They add drugs to the different wells and see if they can inhibit this green color. In a way, you can the cell population of T cell population is reduced. If you see here, finally they could come out with one molecule like 1H1 indole 3-carbaldehyde 8-quinoline hydrazone. It's a molecule which can inhibit the T cell growth. So this T cell growth is comparable when the initial stage is comparable to a cancer cell growth. Okay, if a T cell cancer is there, it divides very fast. So if you can inhibit that with a molecule, this can be directly translated into a novel hit molecule. Then it goes for mouse model and then finally into clinical trials. If the safety it overcomes the safety. Now, if you see, just give a small hint. Okay, a novel hit and it can fetch up to $3 to $10 million. Okay, about 20 to 75 crores is a novel molecule, well-validated molecule. Maybe it can go to preclinical trials, then initial payment you get it. So here is the way for startup, because we have a lot of medicinal herbs or library of known molecules. Novel transgenic lines has to be generated and tested and pull out the very potent molecule which can go into further development, which I'll come in the last. Now, this is an exciting thing, like zebrafish with human cancer gene, what this this is a BRAF one gene, BRAF, which is oncogenic gene, which is present in the human. Okay, so now scientists developed a model and they took this like a homologous gene and replaced with the original BRAF gene of zebrafish. Okay, so now like finally they found out that they could develop a transgenic mix with a tumor, which is very much, which behaves very much like a human tumor, because it is controlled by the expression of BRAF gene of human. Using this, a lot of screening has been done, and finally they could find out that euplomoid, one of the molecule, is very much effective against this tumor, and this has gone up to the clinical trials. Clinical trials of euplomoid combined with BRAF inhibitor for treatment of melanoma are going on. Okay, and also they found out a target. This is the protein target to which this binds and brings in the amelioration of the condition. Okay, so this is one thing. And finally, on oncogenesis, this is also very important. A company called Z Clinics. This is a Spain-based company. I'll just ask you to go through the website of the company. They have a lot of things, anti-inflammatory model, Alzheimer's model, Zio test, cardio test, etc. Okay. Now this is very interesting because they have developed a method for personalized cancer treatment. What they do is they take so how to find the best drug for a cancer patient. You cannot test the lot many drugs to a cancer patient because of our pharmacogenomics. We have different inherent tendency to respond to drugs. So here comes the this thing. They are taken like biopsy from a patient. Okay, then take a few cells and transplant into the zebrafish. Or when the embryo is developing, just inject this fish like a micro injection. When the embryo is developing, inject. They'll go into integrate. Okay, to the organ. So then incubation with the you have to incubate with the different drugs. Okay. Imagine cancer cell mass when it's the beginning, then imaging a cancer cell mass at the end, maybe after 3 days or 4 days. So just different drugs which can reduce the population of this implanted cells, human cells. Like that, they come out with the molecule trusted with drugs which can heavily inhibit this cancer cell, will work in the patients because the cell is originally taken from the patient. This opens up a lot of avenues for personalized cancer treatment. You see, like 100 types of cancers are there. So we need 100 types of models or zebrafish to work with the biopsy and things like that. This is a lot of commercial interest it has. Now, this same company has a model for epilepsy. This is the commercialized zebrafish model for childhood epilepsy that mimics the convulsive behavior and seizures associated with epilepsy. The model has a proven track record in successfully identifying new treatments against epileptic disorders, and already 3 compounds are phase 1 and phase 2 clinical trials. So that's clear because one compound going into clinical trials is a big thing because a lot of money is spent, and only if it goes to clinical trials, and then we know that some molecules will emerge in few years for epilepsy treatment based on zebrafish. Now, another is on kidney regeneration. If you see that, one paper, development of high content assay for kidney progenitor cell expansion in transgenic zebrafish. So they found out that okay, here there is a homeobox protein called LX1. This is a transcription factor, the limb transcription factor, which is expressed only in the kidney field. Okay, so I think next. So, yeah, okay. So here is the picture where they use for high content screening. Okay, if after injury, if you can express this limb homeobox gene, you can very well restore the function of the kidney, okay, or make it regenerate. Okay, so they tested different chemicals and came with the UPDH one chemical which can enhance the expression of limb homeobox gene, thereby regenerating the kidney tubules, etc. So this is one very important study in the kidney regeneration. Just went through. If you see that in the kidney disease in human, there are more than 20 types of kidney disease. Just put the picture. So in a way, we need at least 20 types of transgenic fish to find out novel drugs for kidney disease. This is going to happen, yeah, because now the models are ready, the methodology has been standard. Now more people have to use it, and for each disease, they have to find a novel molecule which can ameliorate the condition. Heart repair. Okay, so it was a work on Leukemia. Recently used to identify drugs that limit cardiotoxicity of chemotherapy agent. Doxorubicin, is a chemotherapeutic. A lot of patients who have cancer, they treat with like doxorubicin, but it gives a lot of cardiomyopathy conditions. No? Okay, same way in zebrafish also, if you treat with like a doxorubicin, it causes pericardial edema, disrupts heart morphology, contractile activity, reduces blood flow, decreases cardiomyocyte. So this is a model to test drugs which can ameliorate this condition, which can further go into preclinical studies and go into human use. So they found out that vasinagin, okay, diphenylurea is as a cardioprotective compound. Okay, the target is also identified. It's a malate dehydrogenase 2 protein molecule is a target. Okay, so they found out this vasinagin also improved heart function in mice treated with the doxorubicin. Then affinity chromatography and inhibitor studies demonstrated inhibits this malate 2 hydrogen. So that's a target. This is a hit molecule. Vasinagin is a hit molecule. This malate dehydrogenase is a target molecule. Okay. Now, this is another study for cardioprotectant. Okay. So same way, zebrafish, and then they found out that a compound SB2, the name of the compound is not given. Okay. So using different screens and cardiac assays, they could find out one molecule which serves as a cardioprotectant. In addition, the same compound rescues contractile abnormalities and conduction abnormalities observed in mouse model. Okay, so same way, what works in zebrafish also works in mouse. If it works in mouse, then it can go to preclinical further studies, toxicity studies, and it can end up in clinical phase. Okay, diabetic. Okay, so you see, zebrafish also has a pancreas. Okay, so in one of the study, Wang et al., they labeled specifically pancreas with a YFP, okay, yellow fluorescent protein, particularly one insulin promoter, because insulin expressed only in the pancreas. With that, they used a YFP and another RFP. Here, you see that, okay, the beta cells and the delta cells, RFP expressed, YFP expressed. Now you can quantify this, okay, how much it is, like if you give a drug, okay, whether this population, beta cell population is increasing. This is the study, okay. You can image it or make a video of it, okay, by giving the drug, because if you see that drug dilution on day 2, they give, okay, and then go for the endpoint analysis of a scanning, whether like this drug has increased the beta cell mass or increased the delta cell mass. So what a technique, no? You can just see under the microscope or image, make a video of it, okay. 100 zebrafish, then you can like average the results and find out these are the small molecules which can increase the beta cell mass, okay, thereby increase the insulin production will be there. And they screened like a lot of molecules, no? Like 24 drugs stimulated these are beta cell proliferation. 18 of these are leads or drugs approved for human use. Okay, in another screen, 883 small molecules identified, 20 lead compounds, okay, belonging to retinochromatic and glucocorticoids, which increases the regeneration of beta cells after beta cell ablation. So powerful model to find anti-diabetic drug screens. Okay, now coming, these are, I told a lot of on hit molecules. Now is coming to target molecules. Okay, novel target, 1 minute, excuse me. If you see that, this is one study which Kendall et al. In 2018. This is actually very interesting to read through, because they found that for a human sarcoma, okay, or rhabdomyosarcoma, which is a cancer of soft tissue or muscles tendon, a fusion of DNA on 2 different chromosomes causes the most aggressive form of rhabdomyosarcoma. The fused DNA products are PAX3 and the 2 gene products, no, protein products found in humans. When they join together, this cancer, this goes off, cancer development. So this group of people, what they do, they took this gene and from the human and put it into a zebrafish. Okay, like GFP expression with the promoter, PAX3 and FOXO1 gene. Then with the transposase, told to like a method of transgenesis, put a transposase mRNA into that, and then made this cells, okay, sarcoma cells into a transgenically labeled GFP. Now we can test. Did one interesting thing was now this sarcoma is GFP labeled. So this you can isolate this molecule, this cells. Okay, only GFP can take out from the total embryo. Then through a fax, fluorescent activated cell sorter of you can just remove these cells. Okay, then find out through a microarray analysis what are the genes being expressed in the cells specifically. Okay, when like a tumor is forming, exactly what type of genes are being expressed. Okay, within only this cell. Okay, so this is what they did. Okay, they generated 4 types of transgenic using injection as controls, and then finally PAX3, FOX1. Okay, then using a microarray studies, they found out what are the genes expressed only when PAX3 is getting expressed. What are the genes expressed only? You can find out 180 genes are in common. 529 genes are expressed only in PAX3, FOXO1 transgenic zebrafish. In that, they found out that one of the protein, HER3, is the fold change or expresses 20 times or 21 times more. So they came to a conclusion with several other studies, validating studies, that this is a target protein which is important for making this rhabdosarcoma this multiply. Okay, so now you have a target. Okay, so this is related to same in humans. You have same protein called HES3, HES3. Okay, now using a zebrafish, they could find out that HER3 is a novel developmental target of human PAX3 FOXO1. So this for this tumor, this is the target. So now you can screen different drugs, okay, which can mitigate the expression of HER3. If it's there, then automatically you can. This they have approved in the paper and the different models. At the mouse model, they approved that. This by down regulation of HER3, they can bring the level of the sarcoma down. So this is a very good example telling that using a zebrafish, even a human target, or for a disease, for even cancer, you can have a target identified in zebrafish, which is one of the very marvelous study. Next, we'll get to like a zebrafish in high throughput. If you see that whatever I told, you can make that totally robotic. Okay, automation technology for dispensing like 3,000, 4,000 chemicals one at a time. Zebrafish can be used high fecundity, then neurobehavioral studies, disease modeling, genetic tractability. You can just change a gene, put in a gene. Okay, lot many things you can put in a zebrafish, and I think 25,000 publications they have in zebrafish, and it's huge and keep on growing. You can see this curve, how much it is growing exponentially. Lot many studies are coming in zebrafish using high throughput, and new drugs are getting developed using zebrafish. Another one interesting aspect which got my attention was, see, this is some scientists from Harvard Hux University, neuroscientists. They put a zebrafish under something called a light sheet microscope. Okay, image the brain of zebra. Imagine this is only a pinhead size, very small. Okay, about 20 you can arrange in a nail. Your nail you can arrange 20 zebrafish like embryos, or more than that, maybe 30 in a nail. You can see how much small it is. Okay, and you can microscopically imagine each cell types. Okay, their behavior, and this zebrafish is kept live. Okay, live in the under, because doesn't need food. It is a yolk. If you have a water medium covered, then it will be there with the agarose, soft agarose embedded. It will sit there for 2 days even for development. So the researchers use high-speed light sheet microscope to image the activity of 80% of the neurons in the brain. So any type of neuroactive drugs you can test. Okay, specifically to neurons. Okay, this is very interesting actually. This light sheet, because confocal has a little limitation. Confocal gives a very clear picture, but if it's embryo is this much big, okay, you can go up to this much level. But this light sheet can cover the entire length of the embryo, different angle, no? Horizontally, top to bottom, horizontally, anywhere you can take thousands of pictures and see for the activity. So just a drug and see for the activity, make a knockout there, and then see whether a drug can rescue this knockout or knockdown. So not many opportunities there. Okay. Now, very interestingly, the FDA and EU, zebrafish assays have been accepted for Food and Drug Administration for toxicity safety assessment for investigating new drug. New drug, as you see the different procedure, the new drug approval, even zebrafish assays are used. Then EU, as a directive of 2010 directive, regulates the zebrafish larvae up to 5 days as classified as in vitro models. So any number you can use that don't warrant much approvals, because this is used as a in vitro model. By this 5 days, you have all the organs developed. So any assay you can do that. So this is also so zebrafish embryo is called as 3R complement, which stands for reduce, refine, and replace the use of animals in toxicity assays. Okay, so the zebrafish can be widely used as per the up to 5 days of days post fertilization. Okay, or 5 days of development you can use the zebrafish. So this all advantages. No? If you go back, cancer models, liver models, then like pancreas models, personalized models. So zebrafish is emerging as one of the top model for drug discovery. Now, entrepreneur prospects. So this is also very important for biotech students. You see that whole market for biotechnology by in another 2050 720 billion US dollars. The market for biotechnology, 1 billion is 7,500 crores. This is global. You don't have to worry about it. It's 55 lakh crores by 2050. It's the market for biotech products, bioinformatics, tissue engineering, regeneration, so many things. Okay, just a little bit of narrowing down. See this research. This, if you see that contract research organizations support big pharma. Okay, big pharma. If CRO is developing a technology, they try to acquire this technology or buy out this company. This is getting globally in a robust way. And I just say this discovery phase. CROs, lot of CROs are working. This is a market share of 1.2 lakh crores globally. Okay. Pre-clinical phase about 75,000 crores. Even it is 1%. Okay. 1% is like 1,200 crores. Is a good market for India if you cover even 1% of this market. Okay. 1%, like 750 crores. Okay. 10 or 20 companies can come up. Okay. In this area, startups. Okay. And this is the market for this contract research organization. It goes up to by 2026, the 90 billion. So 90,926, 90 billion US dollar is the market for CROs, where zebrafish has a segment. Now, what are the different areas entrepreneurship? What is zebrafish embryos? Even having a startup, just supplying zebrafish embryos to 100 organizations can have 5 people can have a job and then start up. Antibodies for zebrafish proteins in high demand, because for so many proteins there are no antibodies in zebrafish. Generating antibodies, 10 antibodies you can have a company. Okay. Zebrafish knockdowns, maybe we need 500 knockdowns, and this is a global market. If you have a zebrafish lab producing knockdown after knockdown for each gene, there will be people demanding, okay, give us so many samples of like maybe 10,000 or 20,000 or 1 lakh embryos with a particular knockdown. So many. Zebrafish preclinical research, so many screenings, toxicity screens, whatever organ network. It's a big area. Okay. Hit discovery. Like if you have some few transgenic zebrafish, you can just screen a lot of medicinal herbs. Okay, which we are doing for infection disease. Screen them, just see a phenotypic change or particularly ameliorating a defective heart on some few molecules. Then hit-based startup. Target discovery. I showed you one example. There are more than maybe 3,000 human proteins are not discovered, not known what are the proteins. We know the genes. Maybe there are 100 good targets in this 3,000 different unknown proteins. Using zebrafish, you can have potentially. I think that India has 100 startups, zebrafish startups by 2025. This is only 6 or 7 points. Maybe I can list 15 or 20 areas where zebrafish can be used for startups. Now, drug discovery startup in India. Okay, so as it is the hit molecules, like a normal target can have 3 to 10 million US dollar value. Okay, then a hit molecule have 10 to like a not sorry, the picture is reversed. Okay, here is a target, here is a hit. Okay, so here like a target can have 75 to 250 crores value. What is the revenue stream of startup if some 2-3 molecule? Upfront payment, licensing payment, R&D payments, milestone payments, royalties. These are the global pharmas looking for novel molecules, novel receptors. If you have like a novel target, I'm sure that one of these people is going to come out with some agreement on these things. Okay, then they look for CROs. Okay, who can work for preclinical studies, etc., hit discovery. They have a lot of millions of compounds. So if you have a zebrafish, one zebrafish maybe for screening, you can have an agreement with them and screen their compounds, and they will pay for you. Well, that is like R&D payments, like research and development payment. Many avenues are there. Okay, so potentially we have like 100 startups in India on zebrafish in drug discovery in few years. I'm sure about that. So to another aspect is like internal organs. So that muscle development. Okay, so like this next generation in therapeutics is 3D printing of organ, which we are also discussing in the BRICS Biomet Consortium, which we are also very interested. Now we need a heart. Okay, our own heart. Like if we have a transplant taken for heart and a heart is ready. Okay, another 70 years, or then maybe I need a heart replantation. Okay, I can need a pancreas. Okay, maybe kidney, and a lot of people in accidents need that. So now, like if you have a 3D printed organ ready, which is compatible from my stem cell, it can be developed. Maybe it take 2 months or 3 months. Then automatically no rejection will be there. Also, like compatibility studies, if we make lot many organs. This is the next generation medical. Like I can tell that it's like it can revolutionize the medical field. People can live longer. Okay. Like people have more money, it can have 3-4 organs ready. Whenever it's damaged, okay, you can replace it. Then it go for another 20 years, again go for replacement. Technology will develop. Okay, this is one area which BRICS Biomet is also interested. And zebrafish gives us a good model to study all this organ development. What are the growth factors important for this each organ development? If we can pull it out, then that's also a big market, biological market. It's just a lot of work has to be done, a lot of team has to be gathered to do all this work. Then our lab one is interested, like now the biologics and biosimilar, it's a cell culture. You use cell culture to produce it. This can be also produced, expressed in transgenic fish. Only thing replaced with this GFP, with the insulin, or maybe a monoclonal antibody, like a heavy chain, short chain, then try to recombine them. You get at a very much cheaper cost, because once this is transgenic, you can produce 10 tons of this fish, and maybe 1%, 2% will be the this therapeutic protein, or 100 kilogram protein. In a 3 months time, you can take it. The only thing downstream processing should be highly standardized. Then this is one expression biotech. We have this firm which will be interested to take up biosimilars production in transgenic fish. Okay, now like. To summarize the 40th slide, we saw what is whole aspect of drug discovery, target to hit, and preclinical. We saw how a transgenic fish can be produced by microinjection into the embryo. We could saw that how T cell growth can be assayed using a transgenic fish, and humanized like a protein expressed in zebrafish, oncogenic protein, how we can screen it. And then finally a target for a sarcoma could be discovered from zebrafish, and how zebrafish can be used as a high throughput screening for a lot of other drug screening, etc. Then what's the scope of startups, or scope of biotechnology, scope of drug discovery, or how much market it has. Okay. And then finally, you can see that as a startup, where you can how is the strategy? We can think about that and trying to come out with some heat and this thing. And who are the people who will buy your product? Okay, here it's a knowledge-based industry. So now, like as a PG students, if you really want to venture here, find a lab where you can do good research on a zebrafish. Take two, three unknown protein, try to express in a zebrafish and find out the therapeutic value of this protein, or find a target for a human sarcoma in the zebrafish. Once you do a PhD, then you can come up with 2, 3 patents, and then that gives the startup. And then even in your MSc, if you started in 2, 3 years, you went through lab, get training, pull people, that smartness is there. Then you can have a startup. It's not a very big game. Okay, so finally, I tell to if you want to be successful, 3 tips for success: read something no one else is reading, think something no one else is thinking, do something no one else is doing. So okay, now people used to say, survival of the fittest. It's not like that. Everyone else fits. It's the survival of the fastest. So another 3 years, 4 years, you want to develop a startup, okay, or read a lot of things, okay. Zebrafish, a lot of videos are there on Transgenic and this thing. Try to get in contact with people. Try to go some training, and then best of luck, and thank you all from Prakash sir. Thank you very much. Hello.
Hello. Yes, sir. So thank you, sir, for the enlightenment and interesting talk. And many of our participants would be having queries and would be happy if you can take their questions. Okay. Now the floor is open for question and answer. So either you can type your questions directly in the Zoom chat window, or you can directly unmute yourself and ask your questions to sir. So until anyone is asking questions, or so I have a query, sir. So we have many repurposable drugs which are being screened for COVID-19. So how far zebrafish is utilized in screening of these drugs?
Can you repeat?
I'm asking, there are many repurposable drugs which for COVID-19, which is currently a very big issue. So in that case, how zebrafish have been successful in screening of these drugs?
Okay, yeah, you need to establish a model of zebrafish infected with COVID-19. Okay, and this I don't know whether it has been done. If you have done that COVID infection, because we have done with bacteria, not with virus. Automatically, if you can see if it mimics the same human condition, I don't know, like in the lungs, because here it's the gills. Okay, so a lot of infection. Then you can try to have a COVID-19 with maybe with a GFP tag. Yeah, COVID-19 with a GFP tag. Infect here and see if your drug can remove the GFP in the zebrafish. That is the model. You can test different drugs for COVID-19 using zebrafish. You got my point?
Yes, sir.
Yeah.
Actually, the zebrafish has 82% similar genes to human beings. So yeah. I think it can be used.
Yeah, it can be used. It can be used. But I think some fluorescent tag should be there. If it COVID, a small fluorescent tag, you can see the multiplication. You can image the multiplication of COVID in zebrafish. Okay. Then test the drug. Okay. If you can bring down the multiplication, then that is a good drug for COVID-19. Small molecules, I mean, small molecules.
And today itself, I was just viewing zebrafish and COVID-19. So I came across a paper in Journal of Pharmacology where researchers have screened for these drugs against COVID-19 using zebrafish disease model.
Okay, good. Okay.
So it has a strong potential.
Yeah, sure. Yeah.
So I would request if anyone has questions, either you can ask your questions directly or unmute yourselves. I have a question, Mr. Kalai Rasi is asking.
Okay.
So kindly unmute yourself and please ask your questions. Ask your question to sir. Okay, so you are not able to unmute yourself. Just 1 minute. Okay.
Hello. Good morning,
Yeah.
sir.
Yeah, good morning. Yeah, tell me.
Sir, I do have a question. Like you are mentioning about the startup ideas. I mean, the areas available with is using zebrafish as a duck target and everything. As we know, for any clinical, researchers, we do need to get certification like GLB certification and other things. For startup using zebrafish, do we need any certification like from ICMR or any other?
Yeah. There are some. There are few because you should clear by ethical committee. Clearance should be obtained. So I don't think it's very cumbersome because it's already many labs are doing. Ethical committee clearance, then you can apply for that. They will allow for any type of zebrafish work.
Okay, sir. Okay, thank you.
So until anyone else is asking the question, in your 18th number slide, you had said about zebrafish and related to cancer, I think. So if you can just open that slide 18, slide number 18, then I had a question.
18? Wait. This one, yeah, 18, yeah.
I think so. Okay, so this ISB or what, and what does it has similarity with the tumor cell?
Yeah, this is the blood vessel, no? This is the zebrafish blood vessel. If you see the picture on the right, same with tumors grow by generating new blood vessels. This is how they grow. They get connected to the nearby arteries and pull in the blood. So here, similar way, this ISP also grows as a blood vessel. So anything inhibiting this ISP growth is anti-tumorigenic compound. You got the point? Because this is labeled with the GFP. If you see the next figure, you can label this GFP and see its development. Okay, when the zebrafish is growing, you can see this ISP formation. So if you can inhibit this ISP formation, any chemical, okay, in a small molecule, then it gets a direct clearing as a drug for cancer, because that can also inhibit this blood vessel formation, no? Somewhere like from this tumor.
Yes, sir.
Got it?
Yes, sir. Got it.
Okay. Okay. I don't think any questions.
So generally people have questions, but they don't ask it. So they can type your questions. You can also, you're free to type your questions and
okay. You can send by mail. You can send it.
Yes,
by
yes,
mail.
yes, that would be fine. Yeah. I would also, you can also email your queries directly to me, and we will compile all the queries and then email it to sir.
Sure.
So, and I have another thing to ask. That is, I read recently that the acceptance of the GM by general public is low. So why is it, sir? Why is the genetically modified fish accepted?
No, because this doesn't go for consumption or anything like that. Okay, this is a tool, laboratory tool, which is for enhancing drug development. Okay, doesn't go to the public. Okay, it's only for medicine, and I don't think any problem will be there. This thing for using transgenic fish.
So, and how would you compare zebrafish with other models like maybe mice, pig, in which we do screening?
Correct. So this, as I put a earlier table, no, first table, I think, first or second table, which shows that in vivo imaging, it's best in zebrafish because it's very transparent. Okay, transparent and very small. Okay, and lot many results come from this in vivo imaging. All organs develop. So if you you want to see one organ, you can just image this organ by keeping the animal live. So any number of small molecules drug can be tested. This is not available in other mouse and rat models. No? You can take an embryo out of the womb and put it under a microscope. Our fly model means it's like it's an invertebrate. No? So zebrafish has this advantage. And also gene modification. Lot many tools have come for gene modification, as shown the examples, which really helps in zebrafish as a tool for drug discovery.
And even the reproduction rate is high, I think that can.
Yeah, reproduction rate is high. Yeah.
Because if we do a experiment in mice, it generally for generating the mice population, it takes many days.
Many years, and also a lot of space.
Yes, and we have to maintain them in sterility, sterile conditions.
Correct. This is much easier, much easier.
Okay, sir. So then I would request all our participants to type your questions and to me by email and send it, and we will compile all your queries and we will send it to sir.
Okay.
So before we wrap up, would you like to say anything to all our participants, sir?
Yeah, that's what this area has got a lot of scope. Those who are really interested, you think about it and read many articles, because reading is very important. Try to understand many things. Maybe you don't understand, but try to understand. By YouTube, there's a lot of information. Gain knowledge, because it's a knowledge-based industry, and whoever is enthusiastic and whoever is as the seed, automatically will be a winner in this area of startups or transgenics, etc. Somewhere you will end up in a good position. Okay.
So thank you, sir. Thank you, Professor Vincent, for enlightening us and informative presentation on transgenic physics in drug discovery and opportunities for entrepreneurship, and valuable insights enabling us to learn how zebrafish serves as a tool for fastening drug discovery to combat any disease and possesses significant entrepreneurial prospects.
Sure.
And on behalf of KIT TBI, kindly accept our sincere thanks.
Okay. Thank you very much. Thank you.
And thank you, sir.
Thank you. Okay. Bye-bye. Bye. Have a nice day.
Before we close today's enlightening interactive webinar, I deeply thank all our participants for joining us today and for their valuable time. And we request you to join us for our upcoming webinars of the Drug Discovery webinar series on 26th and 28th September. So let us all prioritize our health and be safe at home. Thank you. Thank you all.
Thank you.